Assalamu alaikum and good evening. Welcome to the special session of invited speakers. I am Saddam Hussain, the moderator of behalf of organizing committee of Third International Conference on Applied Zoology 2020. Our session will contain the talks of international speakers from multiple fields of zoology and applied zoology. It's a pleasure for me to introduce our first speaker, Professor Dr. Muhammad Jalil Zure Zahra from Islamic Republic of Iran. Professor Dr. Muhammad Jalil Zure Zahra is the Director of Scientific Information and Communication Department. He is well known as an aquatic health and disease specialist. Dr. Zure Zahra is also the member of National Viral Diseases Strategic Network. Currently, Professor Dr. Muhammad Jalil Zure Zahra is working with Iranian Fisheries Science Research Institute, Tehran, Iran. Let's start our session with first presentation of Professor Dr. Muhammad Jalil Zure Zahra with title Recent applications of nanoparticles-based lateral flow immunoassay as a novel diagnostics tool for aquatic animals' infectious diseases. Please, Professor Dr. Muhammad Jalil Zure Zahra from Islamic Republic of Iran.
In the name of Allah, the Compassionate and Merciful. First of all, I would like to appreciate from my dear colleagues in the Third International Conference on Applied Zoology 2020, and also my dear respected colleagues and friends in the Applied Zoological Society of Pakistan and Qaidul Azam University in Islamabad of the Pakistan. I would like to talk about my topic, recent application of nanoparticles based lateral flow immunoassay as a novel diagnostic tool for aquatic animals' infectious disease. My name is Muhammad Jalil Zure Zahra, as an associated professor, and my dear colleagues Dr. Fatemeh Hassan Tabar and Dr. Mina Ziarati and Dr. Fatima Kies. My department would be Scientific Information and Communication in Iranian Fisheries Science Research Institute. Immunoassay chromatographic assay, also called lateral flow, dipstick immunoassay, or simply strip test. Our simple device intended to detect the presence or absence of target analyte in sample matrix without the need for specialized and costly equipment. A widely spread and well-known application is the home pregnancy test for the woman. The liquid sample is dropped on the sample pad. The antigen in the sample forms an immunocomplex with the antibody labeled with colloidal gold. Its complex moves along with the liquid sample and makes a contact with the antibody embolized on the membrane, followed by forming an immunocomplex with the immobilized antibody, resulting in the generating a colored red-purple line. The effect mechanism is very easy, and the lateral flow immunochromatographic device has special important parts. For example, absorbing pad, control line, capture test line, nitrocellulose membrane, gold conjugate pad, sample pad, and silver salt pad. And it would be the same, the typical rapid test lateral flow device. The sample pad and what do you need to know about your sample before? Variability of target molecule concentration. Define sample volume to be applied. Variability of sample composition. For example, pH, sample composition may have to be adjusted by sample pad pretreatment. Sample viscosity limits density of the pad material. Unspecific interaction of your target with the pad material. Define pad blocking requirements. Unspecific interaction with test line reagents may require additional adjustments. Need of retention of particles contained in the sample. For example, red blood cells. The conjugate pad typically is the physically smallest part in the lateral flow test. Fulfills a diversity of functions. Absorb the volume in which the detector conjugate is added to the pad. Does not interact with the conjugate. Maintains the conjugate integrity upon drying. Maintains the conjugate in the dry state. Can easily be more than a year at the real-time PCR. Relays the conjugate easily and completely upon contact with the sample liquid. Allows for interaction between the detector reagent in the conjugate and the target in the sample. The analytical membrane. Typically, this is a large pore size nitrocellulose membrane. The membrane are available in very broad range of sample flow characteristics. All nitrocellulose membrane contain a surfactant, usually an anionic surfactant that makes them hydrophilic. The wick, its task is to soak the sample liquid and all reagent that have not been absorbed at the test and control lines. It must prevent the backflow of this liquid into the drying membrane as long as possible. Select a cotton paper with an absorption capacity that is much higher than the sample volume. An advantage of this method would be can detect antigen or antibody. Commercially available, easy to perform, limited, no instrumentation, single use, rapid test, and user-friendly format. Very short time to get test result. It's very important. And long-term stability over a wide range of climates. Relatively inexpensive to make. And disadvantage would be results are qualitative, not quantitative. Rapid tests can be less sensitive and less accurate with other diagnosis methods, such as real-time PCR. It would be natural. And the type of lateral test format would be strips, cassette, multi-devices, and midstream. For example, for human chorionic gonadotropin and LH only. Let me show you a special clip about how lateral flow immunoassay works. The test line 1, test line 2, control line. It's blood, milk, urine, and other samples which interact in the chamber region. and antibody conjugate, capture antibody in the test. All of the components. And you see the result, positive result and negative. Will it compare together? And let me talk about a very important viral disease in aquatics, viral nervous necrosis, VNN disease. We selected it as a good sample for this rapid diagnosis. Now the virus may enter the fish via the epithelial cells of the intestine, skin or nozzle, and transport to central nervous system via the peripheral nerves like with this epithelium. NNV can be vertically transmitted by carrier broadstock or horizontally transmitted from subclinically infected fish to other susceptible fish. And it would be mechanism of the VNN one-step diagnosis kit. And as you see, has a special region of the T coated with the NNV specific antibody. A region of the C coated with goat anti-rabbit immunoglobulin, and the sample loading chamber. And as you see, the one-step diagnosis kit total procedure, including reading result, is within 15 minutes. This kit is able to detect the head homogenate of naturally NNV affected group larvae with titer of 10 for TCID50 determined in GF1 cell line. And it would be comparison between the rapid diagnosis test with the RT-PCR. And when we have no without any clinical sign of the VNN, maybe you have just 2 from the 4. And in the real time, 4 from the 4. And when the 50% of fish lose appetite, 20% fish swim abnormality and mortality began to appear, you have 4 from 4 positive. Very, very same to the real time. And when mortality stopped and the surviving fish did not show any clinical sign, the accuracy of this rapid test would be decreased. 2 from 4. And so it would be best period when the clinical signs were appeared. And it would be natural and it would be logical matter because real-time PCR, very, very high sensitivity, very, very high accuracy, and maybe very, very, very so expensive. Okay, and we have a special catalog for this diagnosis method, the procedure and the other description of the result. And in the result analysis, you have in the positive control, you have just 2 red color in C control and T in treatment. And when the color would be in the T is weak, maybe the infection is low. And in the negative, you have just red color line in the control line. And in the T, you have not any control, any red color line. And if you have not any red color in the C and in the T region, you must repeat again your examination. And it would be continue more and more. And this is our procedure. And as you see, is an infected golden gray mullet from the Caspian Sea. After the autopsy, we selected 2 special important tissue, brain and eyes. It would be the target tissue for sampling. Our instrument and prepare supernatant preparation in the special Eppendorf tube, and it would be better you will try to centrifuge and put the above water for the examination is better because it's full of the virus. Okay, let me show you the nice clip for finally. Okay, it would be a rapid diagnosis kit animation, and as you see, it would be the procedure of the NNV sample size, preparation of NNV sample after you autopsy of the larva affected, extract the sample, maybe fish eyes or brain, sample extraction, and you put all of the suspected tissue in the special epigraph. It has a special buffer for the solution, and after that you use grained sample, and it would be combination together completely, and after that you use from the special rapid diagnosis kit in the special package. As you see, the nitrocellulose pad, and maybe you mixture some of the sample together and use just 4 drops is enough, and you put in the sample zone 1, 2, 3, and 4 drops would be enough. After that, you wait 15 minutes for the result, and you see the positive and negative. As you see in the negative, just red color in the control, and in the positive you have 2 red color line in the control and in the treatment. Thank you so much for your kind attention. Wish you all the time be safe and healthy in this critical current COVID-19 pandemic. Have a nice time, my friend and my dear respected colleagues. Goodbye.
Thank you, Professor Dr. Muhammad Jalil Zureh Zahra, for your nice presentation. Now, I would like to invite Dr. Meena Ziarti from Department of Microbiology, Johram Branch, Islamic Azad University, Johram, Islamic Republic of Iran. She will present with title Possibility of COVID-19 in Animals and its Control and Prevention. Please, Dr. Mina Ziarti from Islamic Republic of Iran.
Hi everyone, I hope to be safe in COVID-19 situation. I'm Mina and I am PhD in microbiology from Islamic Azad University of Jahra. I'm delighted to be here to make presentation about the possibility of COVID-19 in animals and its control and prevention. For introduction, I'll begin with coronaviruses are a large family of viruses. Some coronaviruses cause cold-like illness in people. Others cause illness in certain type of animals. Some coronaviruses that infect animals can be spread to humans and then spread between people, but it's rare. As we know, latest coronaviruses, so-called COVID-19, and spreads mainly from person to person. To date, animals don't play a significant role in spreading to humans, but infected animals may have potential to spark new outbreaks. In fact, the situation of COVID-19 raises many questions. Can COVID-19 transmit to other animal species? How does COVID-19 behave in other animals? With possible contamination of animals, what control and prevention should be taken? Infected animals with COVID-19. There have been reports of animals being infected with COVID-19, and we know that cat, dog, and some other animals can be infected with COVID-19, but we don't yet understand all of animals that can get infections. So far, large cats like tiger, lion, puma, and pets such as cat and dog infected with COVID-19. It is suspected that they became sick after contact with infected person to COVID-19. On October 2020, a tiger at a New York zoo exhibited some symptoms of COVID-19, such as mild coughing, lethargy, and decrease in appetite. It was the first US case for COVID-19 in animals. We are still learning about it, but it can spread from people to animals in some situations, particularly after close contact with infected people. On April 2020, in the Netherlands farms, first reports of mink contamination with COVID-19 was provided. In these farms, COVID-19 was also approved in some farm workers. The sequencing were related to known human sequences, but separately from mink sequences. The most likely explanation for this is introduction of virus by human and transmission. Transmission category two types, direct and indirect. Direct by infectious droplets of infected animals. Indirect by feed, vomits, bedding. Animal models and at-risk animals. Small animals are essential tools for futures of COVID-19, studying the pathogenesis, transmission, treatment, and something like that. Research in hamster has confirmed COVID-19 spreading between animals. COVID-19 utilized ACE2 as the cell entry receptor, which are abundantly found in the epithelial of humans and animals. Examination on sequences of ACE2 from various animals predicts susceptibility of animals to COVID-19. Animal models of COVID-19 have been reported, including mink, hamster, frogs, bat, marmots, and green monkey. Experimental transmission by direct and indirect contact reported for hamster, frog, and domestic cats. Lastly, unintentional human-to-animal transmission with tiger, dog, and cat. In this slide, you see animal models and at-risk animals for different animals. As you see, cat, frog, hamster, and bat can be infected by COVID-19 and can spread to other animals of the same species. Mink can be infected by COVID-19 and can spread to other animals and likely can spread by two people. Pets and COVID-19. There have been several reports of infected humans spreading the COVID-19 to their pets. It's still not clear how often this happens. Cats and dogs are close contact with humans, and therefore it's very important to understand their susceptibility to COVID-19 for control and prevention. Replication of COVID-19 showed that COVID-19 can effectively infect cats and virus transmission with airborne route. Dogs have low susceptibility. Livestock, including pig, chicken, ducks, not susceptible, and some animals may get sick and show signs, symptoms such as coughing, high fever, loss of appetite, vomiting, and diarrhea. In these circumstances, do not let them lick you and kiss you, share food, and sleep in your bed. On contrary to washing your hands, minimum 20 seconds, avoid coughing and sneezing on your animals, and keep animals indoors all the time. How do you protect your livestock or other farmed animals? People not have access to animals. People with traveling abroad in the last 14 days and people with COVID-19 symptoms. Also, you should monitor pets for any signs of COVID-19 and you should wear gloves and masks for close contact or taking care of animals. Conclusion. Currently, humanity is facing a pandemic of COVID-19. It's spreading between people, also was shown to infect a number of animals under experimental conditions. Researchers worry about it and that virus could end up passing back and forth between animals and people. Based on the limited information available to date, the risk of animals spreading COVID-19 to people is considered to be low. More studies are needed to know and how different animals could be affected by COVID-19. Anyway, protection and control is very important in animals. Thanks for your attention and time. Have a nice day. Good luck.
Thank you, Dr. Mina Ziarti, for your wonderful talk. Our next presentation is by Dr. Mina Ahmadi from Iranian Fisheries Science Research Institute, Natural Resources Research and Education Center, Kerman, Iran. She will present with the title, The Negative Impacts of COVID-19 on Aquatic Processing Industry. Please, Dr. Mina Ahmadi from Iran.
In the name of God, thanks to the organizers of this Congress. My name is Mina Ahmadi. I'm researcher at the National Fish Processing Technology Research Center of Iran. The title of my presentation is The Negative Impacts of COVID-19 on the Aquatic Processing Industry. Introduction. The COVID-19 pandemic will continue to inflict heavy damage on aquatic processing industry, particularly for fresh products and popular restaurant species. On the supply side, fishing fleets are lying idle, and the deteriorating outlook has seen aquaculture producers drastically reduce stocking targets. Many seafood processing businesses had to reduce production or close down completely in response to COVID-19 and lockdown restaurants. It is expected that the production of seafood and its trade value is reduced. Demand for packaged and frozen products has had a significant impact on retail sales as household demand for non-perishable food has increased. The labor shortage caused by the epidemic has affected everything from initial production to the end of the product chain. Furthermore, widespread containment measures can have a notable impact on nations that trade significant amounts of seafood, reducing foreign incomes or threatening food security. Keeping the supply chain open is fundamental to avoid a global food crisis. The COVID-19 pandemic negative consequences. Many fisheries faced complete shutdowns at the onset of social distancing restrictions if they were not considered vital to national food supply systems. Knock-on economic effects from markets disruptions have further impacted small-scale fishers' ability to pursue their livelihoods through twin disaster of reduced demand and attendant collapse of prices. Slashed prices due to reduced demand from local restaurants and hotels have drastically reduced fishing activity, and factories are closed or operating at reduced capacity. Access to cold storage is more important now than ever to reduce fish waste and loss and maintain local food security. Fishing communities and the ports could potentially become hotspots for rapid infection due to the migratory nature of fishers and frequency of international visitors. The COVID-19 pandemic negative consequences continue. In processing plants worldwide, women tend to occupy temporary and lower paid positions. Don't have access to social protections after losing their jobs and are more likely to be laid off. In many places that are highly dependent on tourism, declines in global travel will have devastating impacts on local livelihoods and likely lead to increased pressure on local resources to meet food and livelihood needs. Steep plunge in prices and supply. Shrimp, for example, are selling for a mere 10% of their normal price. Similarly, dramatic price drops have been registered for fresh salmon, trout, and cod. Massive flight cancellations have hurt trade in some high-end fresh seafood products. Fish and seafood exports have fallen by 30% since the outbreak, and Indian fresh consignments to chain have dropped by 10% to 15%. The pandemic is also affecting supply, with economic restrictions keeping many boats anchored in ports. The catch has collapsed by about 80%. The region's confinement measures have hurt small-scale fishers the most, with less than 10% of their vessels currently casing nets. To tourism, which has also been heavily affected by the pandemic, tends to be the only economic alternative for fishers in these countries. Impact of COVID-19 on the market. Fish and aquatic food value chain is currently witnessing a medley of challenges ranging from shutdown of operations, changing consumer demands, market access and logistical problems, and transportation and border restrictions. Supply chain interruptions caused by disruptions in transportation, trade, and labor have halted both fish farming and commercial fishing operations. Delayed stocking of aquaculture feed and system is impacting production, with rising prices threatening to be a key followed closure of restaurants and hotels, which represent large buyers of fish and seafood, has impacted sales significantly. Lower demand, setbacks to exports, and higher costs of operations are chipping off profit margins of fisheries and seafood companies. The entire fishing industry comprising open catch, culturing, processing, preserving, storing, transporting, marketing, and retailing of fish or fish products has been impacted. A few of the market forces at play during the pre-pandemic period included advancements in freezing technology and cold chain logistics, and the ensuing popularity of chilled and frozen seafood. Robust demand for prepared and preserved cured seafood options among consumers on the lookout for safe and convenient food options, storing strong revenue potential of fish meal and fish oil establishments, seafood-only restaurants, and expanding varieties of gourmet seafood dishes, and the healthy growth of farmed seafood in the food service industry. Conclusion. Fish and fish products are a key component to a healthy diet and are safe to eat. Misleading perceptions in some countries have led to decreased consumption of these products. Yet coronavirus can't infect aquatic animals, fine fish, reptile, amphibians, and invertebrates such as crustaceans and mollusks. Therefore, these animals don't play an epidemiological role in spreading COVID-19 to humans. COVID-19 was affecting aquatic food system in two significant aspects, the supply and demand for aquatic food. These two aspects were directly related to food security, so food as well nutritional security is also at risk, as the supply of fish and other aquatic food was disrupted. It created less diversity and availability in the market, the raised market price, so the consumer couldn't get desired fish species and other aquatic food items that put adverse impact on consumer demand and consumption. Also, reduce dietary diversity, as if they buy fish with high price, they couldn't afford more diversified food items for diet that ultimately put adverse impact in nutritional security. Here is reference. Thank you for attention. Again, thanks to the organizer of this Congress. Bye.
Thank you, Dr. Meena Ahmadi, for your wonderful speech. Our next presenter is Dr. Mahmoud Hafizi. The presentation is the collaborative work of Dr. Mahmoud Hafizi and Dr. Shahram Dodgar from Vet Azad University, Tehran, Islamic Republic of Iran. Topic of the presentation is Artemia enrichment for sturgeon fish larvae. Please, Dr. Mahmoud Hafizi from Islamic Republic of Iran.
Hello everybody, I am Mahmoud Hafizieh from Iran, Iranian Fisheries Sciences Research Institute, and my topic is Artemia enrichment for sturgeon fish larvae. This is the Caspian Sea, the biggest lake in the world, which at the north of this sea there is fresh water, but at the northern part of Iran, I mean that the southern part of the Caspian Sea, that is not exactly saline water, but also brackish water. I mean near to 11 to 13 ppt. And we have sturgeon fish, which is very famous in all the world. And you see the pictures of 5 native species: Accipenser and Hoso genus. In this figure you see the CPUE, which showed the fluctuations, but unfortunately you can see the decreasing of this fish catchment because of overfishing, because of lack of effective management, unfortunately pollution and illegal trade. And we have 4 centers for artificial spawning and restocking the sturgeon fingerling directly to the rivers go to the Caspian Sea. You see in 1993 more than 4 million fingerlings, and in 2015 more than 21 million fingerlings released to the Caspian Sea, based on the activity that we have done in these 4 centers at the northern part of Iran. For that cases, we use the broadstock of different species. Mainly in this project we use Accipenser persicus, and you see the artificial breeding, and what is our problem in those 4 centers for restocking. Unfortunately, after fertilization of the eggs and the hatch hatching, we have high mortality of larvae, maybe because these fishes is anadromous. We must release directly to the river, and we have some problem in our rivers at the northern part of Iran. Another is decreased resistance the produce larvae against environmental stress, and decrease of the growth rate in larvae culture period. And why we have some problem? Because we have degradation of their natural habitats, river pollution, drought years, dam construction, and unfortunately bycatch fisheries. As every experts know, for increasing survival and growth rate of the fish, we can do 3 strategies: good management, vaccination, and using the best feed for nutrition. I mean, we must we must know the requirement of the fish nutrition, and we must prepare the fatty acid, vitamins, protein, and so on. Of course, with these 3 strategies, we improve the immune responses and health status, and finally we have good survival and growth performance. And one of the most exciting procedure for improvement the nutrition in the fish larvae, it is used of monopoly of Artemia, but before we use, we can enrich with different matters such as essential nutrient, vitamins, pigments, fatty acid, and something like this. By this procedure, we give a bioencapsulated feed directly to the gastrointestinal of the fish. Okay, what is the objective of our project? It is to determine the effects of different HUFA oil concentration, enrichment period, and vitamin C level on growth, survival rate, salinity tolerance, and vitamin C in Persian sturgeon fish larvae, and the possibility of releasing larvae directly to the Caspian Sea is another objective that we have done in this project. And the experiment is the effect of HUFA oil supplemented with vitamin C levels during 2 enrichment period, growth rate, survival percentage, SGR, FCR, CF, vitamin C accumulation, crude protein, lipid, and fatty acid compositions of Accipense persicus larvae, and finally salinity tolerance in these larvae for 6, 12, and 8 ppt at different hours. As material and methods, first we prepared the Accipense persicus larvae. As a preliminary, we must to hatch the Artemia romiana cyst, enriched these napoli with vitamin C 10, 20, and 30%. In HUFA oil, we use ICES 30/4 and sturgeon ovary oil for feeding Accipense persicus larvae between 4 to 20 days after hatching, and during 12 and 24 hours as enrichment period, and calculation of measurement of vitamin C of Artemia urumiana and Accipense persicus larvae before and after enrichment, which HPLC method, measurement of crude protein by using Kjeldahl method, total lipid using Saxelet method, fatty acids using gas chromatography, total length, total weight, and SGR, FCR, CF, and survival as a percentage in Accipense persicus larvae. And for salinity test, we put the larvae in different saline water such as 6, 12, and 80 ppt. For 6, 12, 24, 48, and 72, and finally 120 hours. And for the analyzing statistically, we use ANOVA with Turkey's multiple range test in 95% confidence interval. Okay, this is the preparation of larvae. This is the salon that we use 3 40 liters tanks for each replication, containing 25 liter well water with 1 liter per minute per tanks as a flow rate. This is the hatching of Artemia oromiana cyst room, and collection of hatched napoli with using the light trap, the counting the napoli, and enrichment. This is the procedure for enrichment of Artemia napoli, one at 12 hours and finally at 24 hours. We use HPLC, Kjeldahl, and Saxelet for measurement of different parameters. Here you see the procedure for larval rearing and monitoring for water quality. Of course, measurement using the devices. And this is our experiment. I means two-way ICES and estrogen ovary oil with different percentage of vitamin C, namely T25 to T36. And in this study we found the main factors, I means the oil, Hufa oil, and vitamin C. An enrichment period had not effects on growth, but the combination of oil sources, vitamin C levels, and enrichment period significantly affected on total length in sturgeon fish larvae. ICES supplemented with 20% vitamin C during 12 hours enriching was the best combination and had significant effects. Oil sources and vitamin C levels significantly affected on FCR in sturgeon fish larvae, but enrichment period and combination of three main factors had not any effects on FCR. Here you see the survival percentage of larvae in 12 ppt salinity stress test. The control after 48 hours completely died but other treatments fortunately have good survival rate even after 120 hours at 12 ppt salinity. This is the figure before salinity test of survival rate of larvae when they enrich with Hufa and vitamin C. There is no any differences between control and other treatments. This is after salinity test in 12 ppt salinity at 120 hours. You see the differences between the main factors here I mean the different oil, different percentage of vitamin C, and different period of enriching. But between the combination also you can see the differences significantly. In the present work, the supplementation of vitamin C as ascorbyl palmitate to the Artemia enrichment had a positive effect on sturgeon larvae survival and salinity tolerance. The maximum survival percentage was obtained in larvae enriched with ICES supplemented with 20% vitamin C at 24 hours. It is almost 91%. All the treatments, including the main factors and combination, increased the tolerance of sturgeon larvae in 12 ppt, which is the same salinity of the Caspian Sea. Approximately in all cases, more than 95% survival were reported after 120 hours, but in salinity at 80 ppt, all sturgeon larvae were dead after 24 hours. In this respect, vitamin C supplementation in live food enrichment can be a practical means to improve survival and stress resistance of sturgeon fish larvae prior to restocking and or transfers to intensive culture. As HUFA enriched Artemia supplemented with vitamin C had a positive effect on vitamin C, crude protein, lipid, and fatty acid accumulation in sturgeon fish larvae. Here the vitamin C content in fishes in different treatment, main factors and combination. You can see all the treatments, combination and factors have significantly increase the vitamin C content compared to control. Here the protein contents. Here you see the lipid contents. Of course, the saturated fatty acid contents and the mono fatty acid contents, arachidonic contents, EPA contents, DHA contents, and the ratio DHA EPA ratio. It is very most important ratio for showing the changes of DHA EPA in the larvae. In some cases, such as ICES with 10% vitamin C after 12 hours enrichment, it is the higher ratio, but the others compared to the control is not so high. This is the omega-3 omega-6 ratio, which is not good compared to the controls. And finally, vitamin C as ascorbic acid is considered to be an essential component in diets for fishes. Fish larvae are particularly sensitive to vitamin C deficiency, and addition of vitamin C to larvae diets improves survival, growth performance, skeleton development, stress resistance, and immune response. We found no relationship between HUFA enrichment and larval survival. Moreover, the survival of sturgeon fish larvae fed Artemia enriched with HUFA supplemented with vitamin C when placed in high salinity such as Caspian Sea water was significantly higher than fish larvae fed Artemia enriched with HUFA only. Then the supplementation of vitamin C as ascorbyl palmitate, which is we used in this procedure to Artemia enrichment, had a positive effect on sturgeon larvae survival and salinity resistance. When sturgeon fish larvae fed HUFA Artemia enriched supplemented with vitamin C The survival rate in laboratory condition and also in high salinity, I mean 12 ppt, were increased significantly due to the effect of optimal level of EPA and DHA. Although DHA contains Artemia aurumina nanoply enriched with ICES and Artemia aurumina enriched with ICES supplemented with vitamin C significantly increased compared to the Artemia control, I mean no enriched. But the DHA amount in the sturgeon fish larvae fed ICES, Artemia enriched, and the fish larvae fed ICES supplemented with vitamin C did not show noticeable increasing compared to the fish larvae fed Artemia non-enriched. EPA content showed increase in fish larvae, but DHA, EPA, and omega-3, omega-6 ratio of sturgeon fish larvae fed ICES Artemia enriched with or without vitamin C also were influenced and decreased compared to the fish larvae fed Artemia non-enriched. Thank you for your attention.
Thank you, Dr. Mahmoud Hafizi, for your wonderful speech. Our next presentation is by Dr. Umar Al-Habib from Department of Biology, College of Science, University of Zakhu, Iraq. Dr. Umar Al-Habib is going to present with the title, The Effect of Iron Nanoparticles on Some Biological and Hematological Parameters in Albino Rats. Please, Dr. Umar Al-Habib from Iraq.
Hi, everybody. I am Professor Omar Al-Habib from College of Science, Nowruz University, Duhok, Iraq. I want to present the lecture entitled The Effect of Iron Nanoparticles on Some Biological and Hematological Parameters in Albino Rats. With respect to the nanoparticles, they are a very small, tiny particles measured on nanoscales. Normally, their external dimension ranging from 1 to 100 nanometer. Nowadays, nanoparticles are widely used in many fields, such as industrial, biomedical, and agricultural field. With respect to the biomedical, it can be used in diagnostic imaging tool, drug delivery system, vaccines, and immunotherapy. With respect to the industrial fields, they have been used as catalyst in the preparation of sensors and photovoltaic devices. Also, it is used in agriculture, in food safety, and in cosmetics. Due to this extensive use of nanoparticles, we can see that they are occupying a great area in global research activity. With respect to the formation of nanoparticles, they are present or naturally or incidental, or it may be manufactured as a material containing these tiny particles. With respect to the state of the nanoparticles, they are either unbound in an unbound state, or an aggregate or agglomerate states. Example of that, we have nanofilms, which representing one dimension, nanofibers and nanotubes, 2 dimensions, and nanoparticles, 3 dimensions nanoparticles. Due to the extensive use and increasing production of nanoparticles, due to the high demand to these tiny particles and rapidly proliferating field of nanomedicine. You can see that this led to the exposure of living organism to these nanoparticles via different routes. Therefore, it's of great importance or great demand to evaluate the toxic effect of iron oxide nanoparticles associated with their uses in multiple applications. Thus, the main aim of the current study was, number one, to study the effect of nanoparticles, different sizes and doses of nanoparticles on performance, food conversion rate, and mortality. And the second part included the study of these nanoparticles on hematological parameters in albino rats, Ratus nervigicus. With respect to the materials and methods, to evaluate the effect of different sizes and doses of iron oxide nanoparticle on performance, food conversion rate, and toxicity, 5 groups of animals were used. Each of them consists of 8 to 10 rats. To perform the whole experiments, 40 to 50 rats, male albino rats were prepared of 8 to 10 weeks old and weighing 150 plus minus 10 grams body weight. The rats were obtained actually from the animal house at the Department of Biology, University of Zagreb. The animals were kept in polypropylene cages, dimensions 30 by 25 by 17 centimeters, acclimated to the laboratory conditions. The animals were with free access to water, standard rat pellet at libitum. With respect to the environmental condition of the rearing room, actually set at 24 degrees centigrade plus minus 2, relative humidity 50 plus minus 10%, and photo period of 12 hours light-dark cycles. The rats were randomly separated or divided into 5 groups, namely the control, then we have AA, AB, BA, and BB. This table actually showing each group, the size and the dose taken. In addition to that, the control group. In control group, there is no addition of nanoparticles. In group AA, nanoparticles of 25 nanometer external dimension and at a concentration of 1 milligram per kg feed. With respect to the second group, AB, 25 and 10 milligram dose. With respect to the BA, 50 nanometer external dimension, dose 1 milligram per kg, and the last group, BB, also the size 50 nanometer, and the dose 10 milligram per kg. With respect to the performance of the living rat, the following parameters were determined: growth rate %, feed conversion ratio or rate, and mortality rate. The second part included the measurement of hematological parameters, including RBC, HB, HTC, MCV, MCH, MCHC, RBC distribution width, platelet count, MPV, white blood cell, lymphocyte, and granulocytes. With respect to the statistical analysis, was performed using GraphPad Prism 8, and the results were expressed as mean plus minus standard error of mean. Statistical differences were determined by Donut test for multiple comparison, and after finding out the p-value by using analysis of variance, the p-value less than 0.05 was considered as statistically significant. With respect to the result, you can see that the effect of different doses and sizes of nanoparticle on the performance of the rat showed that the percent of body weight gain in iron-treated rats were not influenced or not changed. Only the change was mildly and insignificant. P was greater than 0.5 as compared with the control. In addition to that, also during the experimental period of 4 weeks, the animals were like healthy, showed no behavioral change or any sign of toxicity. This is actually representing the growth rate in different groups, and you can see they are more or less very close to the control one, and statistically there were no significant differences between any of the experimental group as compared with the control. With respect to the feed conversion rate or ratio, you can see they showed better, the nanoparticle treated groups, they showed better feed conversion ratio as compared with the control, since there were no significant differences between any of the groups studied with that of the control. Also, no death was recorded during the experimental period of 4 weeks, and all treated groups remained survival in spite of their treatment with iron oxide nanoparticles. This is actually the feed conversion ratio. Again, as we pointed out, there were no any significant differences between them. You can see that the control one, the black one, and the other, they are the experimental one. You can see that slight elevation, mild elevation or increase in the conversion rate, but this increase was not significant from statistical point of view. With respect to the hematological parameters, you observe that all hematological parameters showed mild and non-significant increase in most parameters in all treated groups as compared with the control, except the red blood cell distribution width, which was just significant at the level of 0.5 as compared with the control. So this is actually, these are the parameters we studied. The values were more or less similar, except for the red blood cell width. You can see that P is 0.03. It means that it's less than 0.05, which is the only significant case. The remaining, they showed actually non-significant differences as compared with that of the control. White blood cell and differential count. With respect to the effect of nanoparticles on white blood cell and differential count as they are indicated in the table, we can see that iron nanoparticle in all treated groups except AB, a group slightly and non-significantly reduced the total white blood cell count. This means they were reduced in AA, BA, and BB group. The other group, AB group, which is 25 by 10, the white blood cell count was non-significantly increased. With respect to the platelets were non-significantly increased in their number in all treated groups as compared to the control. Whereas the platelet distribution width and mean platelet volume and large platelet concentration ratio, all these, they showed non-significant decrease in their values as compared with the control group. Here we can see this is the platelet actually showing increase, but non-significant. In the remaining parameters of the platelet parameters, that's distribution width, and mean platelet volume, and large platelet concentration ratio. In all these, we will see that they were decreased, but they were increased, but this increase was significantly non-significant. From the results of the current study, it can be concluded that treatment of rats with different doses and sizes of iron oxide nanoparticles for 4 weeks did not influence the performance, growth rate, and feed conversion rate in these 4 groups as compared with the control, as well as no sign of toxicity was observed. With respect to the hematological parameter, nanoparticles, they produce non-significant increase in RBC count, hemoglobin level, and hematocrit value, as well as both MCH and MCHC. Also, they were non-affected. Also, the total number of white blood cells was not affected, whereas the number of granulocytes was reduced to variable extent. Thank you very much for listening.
Thank you, Dr. Omar Al Habib, for your wonderful presentation. Our next presentation is by Dr. Fatmi Hassan Tabar from Department of Fisheries Sciences, Surrey Agriculture Science and Natural Resource University, Iran. Dr. Fatmi is going to present with title Introduction of Lateral Flow Immunochromatographic Strip Test for Diagnosis of Aquatic Animal Disease. Please, Dr. Fatmi Hassan Tabar from Islamic Republic of Iran.
Hi everyone, welcome to this presentation. My name is Fatima Hassan Tabar from Surrey Agriculture Sciences and Natural Resources University. The title of my lecture is Introduction of Lateral Flow Immunochromatographic Strip Test for Diagnosis of Aquatic Animal Disease. Introduction. Diseases are recognized as one of the major constraints to global aquaculture production and are responsible for the severe impact on both the economic and socio-economic development in many countries of the world. One of the greatest challenges and opportunity for expansion of sustainable aquaculture has been proven to be in managing the health of aquatic organisms. Effective disease control is paramount within aquatic farming system to stop the spread of infection pathogens. Any successful health management program should monitor the health status of fish, identify and manage risk to fish health, reduce exposure to or spread of pathogen, and manage the use of antibiotics and chemicals. Prompt action in the earliest stage of any disease problem can have an enormous impact on the scale of the outbreak. Conventional isolation for the diagnosis of many diseases still remains the method of choice. However, this traditional method trend to be costly, labor intensive, slow, and might not lead to a definitive diagnosis, even when complemented with histological evidence. In addition to in addition to the traditional techniques used in fish disease diagnostic, a few modern methods like LAMP, real-time PCR, lateral flow, and microtechnology assay are highlights. Okay, the lateral flow immunochromatographic assay, also known as lateral flow test, is a solid phase immunoassay combining the principle of thin layer chromatography and immune recognition reaction. These assays have been widely used to diagnose infection in animals and humans because they allow rapid and sensitive detection of pathogen. The assay is referred to as a lateral flow immunoassay because it is based on the flow of the infected sample along a cellulose membrane by capillary action. It is also referred to as gold immunochromatography assay due to the application of a colloidal gold label antigen or antibody as a detector. Colloidal gold nanoparticles are the most commonly used label in the liver because of their unique features include environment-friendly preparation, high affinity toward protein and biomolecule, enhanced stability, exceptionally higher values for charge transfer, and good optical signaling. Lateral flow immunoassay have achieved broad penetration in a variety of markets. Figure 1 lists the market segments in which lateral flow immunoassays are already in production or are known to be in development. The manufacturing of lateral flow assays that meet the requirement of some of these market segments. So far, lateral flow assay based on gold colloidal particles has been demonstrated to be potentially useful for detecting virus, bacteria, drugs, hormones, environmental pollutants, pesticides, and etc. As shown in Figure 2, a typical lateral flow assay consists of four key components. The first component is sample application pads. Its function is to transfer the sample to other components of lateral flow test strip. The second pad is a conjugate release pad. It is the place where labeled biorecognition molecules are dispensed. The third is nitrocellulose membrane. It's highly critical in determining sensitivity of lateral flow assay. Nitrocellulose membrane are available in different grades. Test and control lines are drawn over this piece of membrane. And the fourth is a wicking or absorbent pad. It works as sink at the end of the strip. It also helps in maintain flow rates of the liquid over the membrane and stop back flow of the sample. Finally, all of these components are fixed or mounted over a backing card. Okay, this technology has many advantages over traditional immunoassay. The assay is simple to use, very rapid, and low cost, does not require a skilled operator or expensive equipment and has high sensitivity and specificity. Evaluation of the result is performed by the naked eye and total assay time is less than 15 minutes. In addition, the kits are stable at room temperature. This, so, these assets make the assay very suitable for on-site sampling to detect pathogen. Several formats have been described for lateral flow assay. Actually, these formats are chosen depending on the analyte. In here, we are described two common format actually. The first is sandwich format. In a typically format, label like enzyme or nanoparticles coated antibody is immobilized at a conjugate pad. This is a temporary absorption which can be flushed away by flow of any buffer solution. A primary antibody against target analyte is immobilized over test line. A secondary antibody or probe against label conjugate antibody is immobilized at control zone. Sample containing the analyte is applied to the sample application pad and it subsequently migrates to the other parts of the strip. At conjugate pad, target analyte is captured by the immobilized label antibody conjugate and result in the formation of labeled antibody conjugate and analyte complex. So these complex now reach at nitrocellulose membrane and move under capillary action. At test line, label antibody conjugate analyte complex is captured by another antibody which is primary to the analyte. Analyte becomes a sandwich between label and primary antibodies, forming label antibody conjugate analyte primary antibody complex. Excess labeled antibody conjugate will be captured at control zone by secondary antibody. Buffer or excess solution goes to absorption pad. And finally, intensity of color at test line corresponds to the amount of target analyte and is measured with an optical strip reader or visually inspect. Appearance of color at the control line ensure that a strip is functioning properly. In figure 1 shows the schematic of general sandwich format of this strip. The second format is competitive. Such a format suits best for low molecule weight compounds which cannot bind to antibody simultaneously. Absence of color at test line is the indication for the presence of analyte, while appearance of color both at test line and control line indicate a negative result. Competitive format has two layouts. In the first layout, solution containing target analyte is applied onto the sample application pad and prefix labeled biomolecule conjugate get hydrate and start flowing with moving liquid. Test line contains pre-immobilized antigen which bind specifically to label conjugate. Control line contain pre-immobilized secondary antibody which has the ability to bind with label antibody conjugate. When liquid sample reach at the test line, pre-immobilized antigen will bind to the label conjugate in case target analyte in sample solution is absent or present in such a low quantity that some sites of label antibody conjugate were weakened. Antigen in the sample solution and the one which is immobilized at test line of strip compete to bind with label conjugate. In another layout, label analyte conjugate is dispensed at a conjugate pad, while a primary antibody to analyte is dispensed at test line. After application of analyte solution, competition takes place between analyte and label analyte to bind with primary antibody at test line. Please see the figure, it shows the competitive format. In conclusion, the immunochromatography tests are now being developed and show great potential for the future. Commercial rapid kits are already available to detect white spot syndrome virus in shrimp and infectious salmon anemia virus in fish. The application of antibody-based method and molecule technology can be extremely used for epidemiological studies. For example, such reach on ISAV highlighted mechanism by which the incidence of ISAV might be reduced. This included the screening of brood fish for ISAV carriage, a screening of a smoke before sea launching, stamping out of net pen before sea launching, and the removal of wild salmonid brown marine farm with SAV before emptying the affected farms. So thank you for your attention.
Thank you, Dr. Fatmi Hassan Tabar, for your informative presentation. Our next talk is by Dr. Mahmoud Hafizi. The presentation is a collaborative work of Dr. Mahmoud Hafizi and Dr. Shahram Dajgar from Vet Azad University, Iran. This will be our last presentation of the session as well as the last presentation of day 1 of 3rd International Conference on Applied Zoology 2020. The topic of the presentation is dried brown seaweed as natural biosorbent of phosphorus of aqueous solution at laboratory condition. Please, Dr. Mahmoud Hafizi from Islamic Republic of Iran.
Hello again. The other topics is dried brown seaweed as a natural bioabsorbent of phosphorus of aqueous solution at laboratory condition. I am Mahmoud Hafizi from Iranian Fisheries Sciences Research Institute with our colleagues Dr. Shahram Dadgar and Ms. Masumeh Hafizieh. This is the world capture fisheries and aquaculture production from 1950 to 2018. You see the increasing of aquaculture activities in the world, and this is the prediction of global seafood consumption 2030. In the future, the farmed fish raised compared to wild cows. Okay, what is our problem? When we have done aquaculture, we produce the waste. Of course, aquaculture is source of significant amount of the waste, which generally leads to deterioration of water quality, and of course removal of two important ions, I mean nitrogen and phosphorus, from aquaculture wastewater is an important environmental challenges. Okay, we have very nice picture from rainbow trout in our country, but for 1 kilogram of fish production, we release a different percentage of nitrogen and phosphorus, as you can see in this picture. If you fed the fish with 100% nitrogen input, 52% in fish body accumulation, 30% in case of dissolved excretion, and 80% as solid discharges. And if you fed 100 phosphorus input, we are feed to the fish near to 33% in fish body accumulated, 21% in case of dissolved excretion, and 46% as solid discharges. In Iran, means in 2019, the total production in aquaculture in fish farm more than 520,000 tons, and actually the trout fish production more than 170,000 tons. Each ton of trout fish products released 150 to 350 kilograms solid dry weight. Nitrogen and phosphorus production per 1 ton trout fish is 7.5 and 45 kilogram respectively. Many aquaculture systems generate high amount of wastewater containing compounds such as suspended solid, total nitrogen, and total phosphorus. Today, aquaculture is imperative because fish demand is increasing. However, the load of waste is indirectly proportional to the fish production. Therefore, it is necessary to develop more intensive fish culture with efficient system for wastewater treatment. A number of physical, chemical, and biological methods used in conventional wastewater treatment have been applied in aquaculture systems. These are the ways for removing nitrogen and phosphorus: ion changes, reverse osmotic, electrodialysis, biological methods, chemical methods, and surface absorption. But in this project we use a kind of biological method which is economical and environmental friendly. This is the system for removing the wastewater nutrient, and as biological method we use seaweed as bioabsorbent, natural absorbent. As you know, the seaweed has so many importance as a human consumption, animal feed, fertilizer, cosmetic industry, green energy sources, and wastewater treatment. There are human consumption, animal feed, fertilizer used in cosmetic industry, biofuel, and finally wastewater treatment. This is the table of economic value of seaweed used for industrial consumption in different products, carrageenan, alginate, the hydrocolate from different kind of species, and this is the products in metric ton in USA per metric ton as $1, and what is our justification? We have near to 3,000 kilometer Iranian coastal line at the Persian Gulf and Oman Sea, and more than 150 different species, the seaweeds, and more than 10,000 metric ton seaweed biomass, which is 2,000 metric ton, especially sargassum, set back from the sea annually. Here's the map, satellite map of Iran, and here you see the coastal line of Sistan and Balochistan, and this is the map of Persian Gulf and Oman Sea in Sistan and Balochistan provinces. This is the seaweed taxonomy, and this is the different pictures of different species of seaweed from Oman Sea: Ulva, Colpomania, Sargassum, Gracilaria, Hypnea, and other genus, Turbinaria, Gracilaria, Colperia, and others. And this is the setback seaweed from the Oman Sea to the coastal line. This is the huge amount of Sargassum erysiphorium, near to 2,000 metric ton based on Ajdari et al. 2007, the stock estimation, and other stock estimation near to 500 metric tons. As material and method, the preparation and characterization of biosorbent, we collected fresh sample of seaweed, Sargassum erysiphorium, from the coastal line of the Chabahar Bay, washing and dried and crushed to appropriate size, and after crushing, the seaweed's particle were passed through meshes with appropriate size to obtain particle, I mean 0.5, 2 and 5 millimeters, in order to determine the functional group responsible for bioabsorbent. The IR spectroscopy was performed. To this, 0.5 gram of seaweed was mixed with KBr4 FTIR spectral analysis. A scanning electron microscope was used to investigate the morphology of bioabsorbent. The isotherm studies were performed in the solution with initial concentration ranging from 0.015 to 0.45 milligram per liter of phosphorus as optimum pH value for ion. After shaking the flask containing the mixture of biomass and solution for 60 minutes, the amount of phosphorus in filtered solution were analyzed. The bioabsorption equilibrium uptake capacity for each sample was calculated according to mass balance on the ion expressed in this equation. This is all other formula that we use for calculation of some factors, and as an experiment set up on absorption of nutrient by seaweeds. In the present study, the effects of several levels of pH 3.5, 6, 7.5 and 9, and finally 10, pH, initial phosphorus concentration 0.015, 0.15, 0.30 and 0.45 milligram per liter, contact time 7, 15, 30 and 60 minutes, and particle size 0.5, 2 and 5 millimeter, and the seaweed particle concentration 10, 20 and 40 gram per liter were studied. You see the procedure for doing well, and the statistical analysis. We use ANOVA analysis after a Shapiro-Wilk test for normalize the distribution data to determine the statistical weight of pH, initial P concentration, contact type, particle size, and seaweed particle concentration. In P removal, regression analysis was performed, and beta coefficient was used to determine the factor weight. Data are present as minimum plus minus ST. All analyses were performed in SPSS version 22. As a result, This is the FTIR spectroscopy of seaweed. You see the fluctuation of different groups. For example, the OH groups along with the carboxyl group confirm the presence of carboxylic acid group in the absorbent. The presence of NH group along with carboxyl group might be attributed to the presence of amino acid group in absorbent. And this is the assignment of bands to functional group on the surface of the seaweed observed from FTIR spectroscopy. Okay, this is the SEM, the micrograph of SEM analysis of seaweed before and after absorption are presented in this figure. The micrograph clearly revealed the surface texture and morphology of the bioabsorbent with a large surface area. And this is the Langmuir isotherm of phosphorus equilibrium. Of course, this is the Freundlich isotherm of phosphorus. In this case, the R2 is equal to 0.14, but in this case R2 equal to 0.8. This is the parameters of isotherm models for adsorption of phosphorus using seaweed. Compare the Langmuir parameters and Freundlich parameters, and this is the graph for effect of initial pH solution in phosphorus absorption of the seaweed. The minimum in 3.5 and the maximum between 6 to 10 pH solution. This is the absorption as a percentage in different time, 7 minutes, 50, 30 and 60 minutes. The highest absorption percentage was obtained in 60 minutes. The initial phosphorus concentration significantly affected phosphorus removal from medium, as the lowest removal was related to the lowest initial phosphorus concentration. In this figure you see absorption percentage based on initial phosphorus concentration: 0.015, 0.3 and 0.45 milligram per liter. The highest was obtained between 0.52 0.45 milligram per liter. And seaweed amount significantly affected phosphorus removal from medium, as the lowest removal was related to 10 gram per liter seaweed concentration. However, there was no significant differences in P removal among 20-40 gram per liter seaweed concentrations. In this case, seaweed particle size significantly affected P removal, as decrease in particle size from 5 to 0.5 millimeter resulted in increase in P absorption from 85 to 98%. And the regression analysis results are presented in this table. The model was capable to predict P removal using the predictors. The highest weight for P removal was related to seaweed particle size and seaweed concentration. The lowest weight was related to medium pH. As a conclusion, the present observation inferred that the Sargassum ilicifolium, as a brown seaweed, we have a huge mainly in the Oman Sea coastal line, showed a significant removal in nutrient, nitrogen and phosphorus Hence, this species can be considered as a potential candidate for bioremediation, domestic and industrial effluent which can cause environmental pollution. It is concluded that brown seaweed species not only can be cultivated in shallow coastal ponds used as bioremediant agent in the effluent treatment plants for conservation of nature and culture and natural resources, especially marine shrimp and fish aquaculture activities, but also after harvesting and drying also can be as a good bioabsorbent. Furthermore, the effluent cultivated seaweeds can be utilized as a feedstock for production of biofuel, biofertilizer, and additives for animal feed, which can reduce the cost of wastewater treatment procedure. This is the picture of Sargassum illicifolium, and thank you for your attention.
Thank you, Dr. Mahmoud Hafizi, for sharing such wonderful information with us. Keeping the time in mind, our special session is going towards its end. I am thankful to all the researchers, scholars, faculty members, and viewers for your participation. With the end of this session, all the activities, programs, and sessions of Day 1 of 3rd International Conference on Applied Zoology 2020 will also complete. Our next and last day of the conference will start at 8 a.m. Pakistan Standard Times. Till that, stay happy, stay tuned. See you tomorrow at 8 a.m. Allah Hafiz and goodbye.
Assalamu alaikum. Assalamu alaikum, dear all participants from all over the world. Just at the end of this session, I would like to give special attributes to our invited speaker from Iran, Iraq. So, firstly, I would like to appreciate very interesting talk by Dr. Jalil, Dr. Zyarti. You both have make our day. Our students like your talks too much. Also, Dr. Amina Hamdi, your talk was very interesting. Dr. Fatima Hassan, Dr. Muhammad Hafizi. You all have done a very good job. And I also would like to appreciate Professor Dr. Umar Al Habib from Iraq. Thank you, Professor, for joining us. We really appreciate all of you. And at the end, I would like to appreciate and I would like also want to appreciate all of our organizing members. You have all of you have done a wonderful job. I would like to appreciate in the presence in the today's opening ceremony, Professor Dr. Muhammad Ali, Vice Chancellor, Quaid-e-Azam University, Islamabad, Professor Dr. Shahid Kamal, Vice Chancellor, Government College University, Faisalabad, Professor Dr. Farhad Jabi, Dean Faculty of Life Sciences, and Professor Dr. Taiba Sultana, Chairperson, Department of Zoology and General Secretary of AZSP, Professor Dr. Salma Sultana, the ICAS 2020, and Joint Secretary of AZSP, and all other colleagues, Dr. Shabana, Associate Professor, Department of Zoology, Government College University, Faisalabad, for her admiring motivation to the students to keep them joining till the end of the session, and also other my all respected colleagues who did the very best efforts. I really highly appreciate all of you. And also at the end, I would like to give my special appreciation and very big applause to the team organizing member of local organizer from Quaid-e-Azam University, Islamabad. So that's all for today. I hope you have enjoyed and have a wonderful time with us. We really feel pleased at the end of these sessions, all the sessions. We have finished with a very good success, I hope. And you will have enjoyed a very good talks, also from national and as well as international speakers. So I hope that you all will be the part of tomorrow. We have a very good start from the morning. We have a plenary sessions, and we have a talks from Turkey. We have from UK and from Italy, and also from the Chile. So you will have a very good morning, but you have to wake up early at 8 o'clock. So we are going to start the plenary sessions at 8 a.m. tomorrow, and other sessions, and that day is half day. So we are going to end. We have a closing ceremony then at 3 p.m. until after that we are going to end our ICAS 2020. So thank